Hello everybody. Let me just start by introducing myself. I'm Michael Fitzsimons, the User Services Manager at the GDC, and also with me today is my colleague, the lead bioinformatician of the GDC, Zhenyu Zhang. So just some ground rules before we get started with the workshop is that you will all be muted by default, just to preserve the audio. However, once the presentation is over, I'm happy to answer any questions you have that you can give me via the chat screen. So just reserve your questions for the end, and I'm happy to go through them all as time allows. Okay, so what are we going to cover specifically today? The goal here is to introduce everybody to the GDC and some of the data it contains and how to access it. And in order to do that, what we're going to do today is investigate some common mutations in colon cancer using the GDC tools. We're going to give a general overview of the GDC data portal, and then we're going to focus on a new application that the GDC just released, the GDC Bio Portal, to locate mutations in tumor suppressor gene TP53. Then we will explore the raw data by downloading reads from the data portal. And lastly, we will investigate the impact of this mutation on gene expression. Okay, so let me just start at the GDC Home Page, just to orient everyone on how to find these tools. So there's a lot of information here generally about the GDC. We're going to focus on the portal today. So this is the interface that most people will use to interact with the GDC. We do have an API for programmatic access, so those who want that access, it's there. We'll talk a little bit about it today, but it won't be a focus of the discussion. If you want more information on any of these things that we're going to be covering today, I highly recommend visiting our documentation site, which has a lot more information on all the things we're going to be covering. Okay, so just a little bit more detail on the data portal. It provides simple and, we hope, intuitive access for filtering your data and choosing which data you want to download. On the left, you can see many ways in which you can filter your data by program, primary site, as well as several types of clinical data. If you want to filter your data by file type, by experimental strategy, data format, options like that you would do on this page. And we'll get more into that in a little bit, but I just wanted to give you a quick overview. So this is a great way to download the raw data, you know, the VCFs, the maps, the BAMs. However, if you want a visual interface, sorry, I'm getting some information that some people may not be able to hear. I believe that others are able to hear, however, so I think it is just a local issue. The main portal is a way to get access to the raw data. But if you want to focus on specific variants and explore on that level, I believe the best way to do that, and the way I would recommend, is the use of our new application, the GDC Bio Portal. So just a little overview here. This is a GDC-specific instance of the Bio Portal, which means it's a little different than the Bio Portal you might be familiar with. We don't have some of the options that you would find in that version of the Bio Portal. Really, our version focuses on gathering data from our open access math files. So really, it just has that information. It provides quite a few different graphics and visualizations that you will see, but you're not able to then look at RNA-Seq or copy number variation or some other options that are in the primary C Bio Portal site. So back to the goal of this study, is I'm interested in looking for variants that are impacting TP53 in colon cancer. So you can see here that this is broken out by TCGA project. We have 4 different variant calling pipelines that we've run, so you can look at the results of each of those. We're just going to jump down to the TCGA COAD project, and we're going to look at the MUTEC2 pipeline today. So I'm interested in looking at variants that are impacting TP53. Okay, so much like C Bio Portal, we do have OncoPrint and some other different graphics, which are all very interesting and informative, and I recommend that you all explore them. Today we're just going to skip ahead to this mutation section so we can see the frequency of the different mutations impacting TP53. This is what is known as a lollipop plot, and as you can see, there are quite a few mutations in TP53, but there's one that is much more common than others, and that is this missense mutation R175H. When I click that, what that allows me to do is to look at just those individuals that are being impacted by that particular mutation. Okay, so maybe what you want to do at this point is you'd like to get access to the raw. Maybe you'd like to explore the individual reads that are underlying this mutation call. So what you could do then is I would go back to the data portal and then download the individual reads to explore in a genome browser. The way I would do that is employ some of the facet filters here. We're interested in BAM files. We're interested in WXS data. This is the whole exome sequencing data, what the C Bio Portal results is based upon. So we're going to focus on that. On the case filter, you're able to sort by what type of sample we're interested in tumor samples. And then we're also interested in a particular individual. So we'll perform that search here in this case field. Okay, so now you can see here that we're down to a single case and a single file. Before we get into this file, let me just highlight a few other things that you can do in the data portal. So if we go to that individual, what you'll see here is some of the clinical information that we have for this individual. You can see some demographic, diagnosis information, some exposure information depending on what was recorded for this individual, as well as the bio specimen information for that case. If you want additional clinical information, that is available in this XML file downloadable here. Okay, so let's go back to finding the reads for this individual. Here's the BAM file. And now you could download this, this 27 gigabyte file, but that's rather large. Instead, what I would recommend is that you download or use our BAM slicing option. What this allows you to do is to specify a region in the genome and download just those reads that span that region. So what I didn't mention earlier is that I'm logged into the system already and I have access to this. However, BAM files are considered controlled access or protected data, so you do need to have access to a particular project in order to download controlled files for that project. So here's where you would specify the chromosome and positions you're interested in. This is the region that spans TP53. Okay, so then we just download that short region of a BAM file rather than the very large entire BAM file. So I showed that in the portal, I'd also like to give you a quick insight into the API and how you might do such a call here. This is an API request. What I'm providing is my token, which I downloaded and put into memory. And then you can also see here that I'm using a BAM slicing endpoint and specifying the unique identifier for this file. So every file in the GDC and every node has a UUID, which is what this is representing. Then I'm also specifying the chromosome number and start and stop site, just as I did in the data portal. So the next thing we want to do is to take a look at these reads in a genome browser. I'm going to use IGV, but you could use a different one as well. To do that, we need to create a BAM index file, so I'm just going to do that quickly using a samtools command. All right, let's jump to IGV. We're going to load that short BAM file that we just created. So here we're showing the whole genome. We need to focus in on the region we're interested in. As you can see, if you zoom out, we have just the reads for the gene, and there doesn't seem to be anything unusual going on here. There's no reason to suspect that just based on this, that there's concern this variant is an artifact of some sort. So just to return to GDC Bio Portal briefly, for this particular individual, here it is, we have an allele frequency about 50 or 0.47. And in the browser, that seems consistent with what we see. All right, so we've looked at the reads. Maybe another thing that would be interesting to do as a researcher is to see how this impacts RNA-Seq results. So to do that, what I'm going to do is I'm going to look at these individuals who are suffering from this particular mutation of interest, this R175H. And so to do that, we're going to go back to the portal and change the filter slightly such that we download data relevant to RNA-Seq results. All right, so back to the facet filters. For experimental strategy, we're going to go with RNA-Seq this time. We have a couple of different ways of counting gene transcripts or reads. We're going to look at just standard FPKM today. And then we're interested in the primary tumor results. So previously I showed you how to specify a single case, but if you have multiple cases, the best way to do that is through the advanced search field on top. This is a discoverable or self-discoverable field or search bar, so if you start typing, it should try to complete it. We're going to search by submitter ID. And here we're going to specify the list of cases that are just what I picked up from GDC Bio Portal. So let's do that. All right. So now we're down to just the RNA-Seq files, just the FPKM results that relate to those particular cases. So if you wanted to download these particular files, the way I would go about that is to add the files to your cart. Go onto your cart. And then you could download each of these individually, but that isn't the recommended practice. We have a command line tool for downloading mini files or large files, and that would really be the best way to go about it in this case. So what would do is to create a manifest, which is really just a list of files that our download tool uses. And I'll show you how to set that up in a moment. I also wanted to demonstrate, in this case, so you created the manifest in the portal. But again, let's take a brief detour over to the API and show you how you might specify that here. So with the API, it's going to look fairly similar. We're going to create a little file that we're going to post to the API. And so if you want more information, again, on the API, there's quite a lot of it on our doc site, but this is the type of thing you would use for specifying filters. It's this JSON object. This has basically the same information that we specified in the portal. It has the cases. You're saying you only want files related to the experimental strategy RNA-Seq and FPKM, primary tumor, and then furthermore, what we're saying is we want it in the form of a manifest. So you can pipe it directly into the GDC client. All right, so I'm going to create that manifest based on that file. This is a similar request to what I showed earlier. We now just created this little file called the mutation manifest, which is identical to the one we downloaded from the portal. So you don't have to use the API, but some people will find it is helpful to go through that. All right, so I found my mutated RNA-Seq results. And now I'd like to plot these. I'd like to compare these, though, with the wild type. So what I would do is go through the same process I went before, getting the results from GDC Bio Portal and then downloading the results from the system. So what we have here on the left is the expression of the wild type samples. And then on the right, we have the expression for just those missense mutations that we highlighted on GDC Bio Portal. And you can see, if anything, possibly the expression trends up, not down. But maybe that is reasonable. It is a missense mutation. There is no real a priori expectation that this would interrupt expression. But let's investigate that a little further. Are there other types of mutations that do interrupt expression? We had been focusing on or filtering for were those mutations that were this particular amino acid change. But if we look at just the truncating mutations, so go through the same process either in the portal or the API, download those RNA-Seq files, and what kind of results do you get? So here are my results I showed earlier. And now look, with this new type, this new class of mutants, you do in fact see a decrease in expression. These are confidence intervals, so you can see that it's a statistically significant impact on expression. Okay, so that's really the first part of the workshop, which is to go over that case study. And before I turn it over to questions, I'll just highlight that there are these different references available. If you want to reproduce this or access any more information, this is a great resource. Generally, if you have any questions at all about the GDC or about the data shown here, I think a great resource would be to contact the GDC Help Desk, and you can reach us here. So with that, let me take a look at the chat screen and see if there are any questions. Can you filter by case having family history information? The answer to that is yes. So let's jump back over to the data portal and check out the different filters that are possible. So we have some clinical filters by default down here, but they don't include everything. So let me jump back to one of those cases and you can see what is possible. I'm just choosing at random here. There isn't indexed data for this particular case, so it depends on the project. All these projects are different. I don't see any here, but often there will be additional information in the clinical XMLs. This will not be filterable in the portal, but it is there. So if a project has the data and it has been indexed, you can search by it. The only thing I'll add to that is that the GDC is in the process of indexing additional fields from those XMLs. So there may be more ways to filter in the future. All right, so another question is, can you BAM slice across many BAMs at once? That is a good question, and we have received that before. I think it would be a nice feature for us to roll out in the future, and we are investigating doing that. For the moment, however, it's something you'll have to script on your end in order to BAM slice multiple file endpoints. Okay, how to identify paired samples? I mean normal and tumor samples from the same donor. So as I said, there's a couple of ways you can get at that information. Often the API is really kind of a slick way to do that, and I'm happy to help you offline in order to get that information easily. The other way, just if you want to add a filter by the different sample types, then you would do this here. But as I said, the API will allow you to output clinical data in exactly the format you want. So it can be very easy to do that. Another option is to access the original biotab files, which I didn't go into. But so this is the GDC data portal, meaning it has the harmonized data. So this is data that the GDC has realigned and recalled variants for and regenerated expression data. Some of the original TCGA data is present in what we call the legacy archive. So this is really, the same filters are possible, but these are the original BAM files and all the original files that were in CG Hub and the DCC. So if there's a file or file type or a particular file, then this would be a great resource for finding that information. I don't see any new questions. I'll give it another couple of minutes to see if anyone has any others.
Michael, we had lots of questions about whether we will be providing slides or the video. We will be providing the video on the GDC website in the future, and we'll send out a notice about that.
Yes, thank you, Sharon. Right, this has been recorded, and we will provide the slides that showed, as well as the link to the video on the support page. I'll just show you where that is. So here's the main website. Here's the support page, which has lots of useful information, including information about the workshops that you're attending. So we're going to put the information, the downloadable file here. All right, so it looks like there are some more questions now. So somebody wanted more information about how the data were processed. That's a great question. So that is all online. There's a couple of different places where you can access that. The really detailed information is on the docs page, and where we go into each of the different pipelines, we're just going to choose mRNA-Seq in this case. And so this has some kind of general overview workflow diagrams, but then we also go into the exact flags that were used for each of the command lines, so that all of the information should be in here for you to replicate the pipelines and the analyses that were done by the GDC. Another question. Will programs like Target get their specimen and clinical data converted to TSV from Excel? So yes, Target, as the user knows, generally has their data in spreadsheets or Excel format. We do also index that. We're actually going through and updating indexing for some of the Target data right now, but there is quite a bit of information already there. So I encourage the user to go into Target and see the different clinical data that is already being indexed. There was a question about mapping file names back to the patient names. It is true that the file name will not necessarily contain the patient barcode, and if this particular user wants help with that, please email a support email and we can help to guide you through how to make that mapping. And then another question about adding additional filters. So I did briefly mention that there's only a subset of data filterable here, but there is more clinical data and biospecimen filters that are possible. So you can add additional filters based on anything that's been indexed by the GDC. We do have different filters that you can add on here. I wouldn't choose this one, I guess, but there are some other biospecimen fields that are available. Another question. Using GDC Bio Portal to figure out which exon and mutation is located. I think that might be best handled offline. A good resource for GDC Bio Portal is the primary GDC Bio Portal's website. They have some tutorials and some additional documentation there. We aren't adding too much documentation on top of that, so that might be the best resource, although the support team here is happy to help you try to find that information too. Then there's a question, do you have plans to include copy number and expression data in GDC Bio Portal? We do have plans to add additional visualizations. It probably will not be through the GDC Bio Portal interface. The GDC is going to be rolling out additional visualization options that aren't going to be based on GDC Bio Portal. They will have many of the same functions as what you've used in GDC Bio Portal in the past, however. That type of information will be available, but it likely will not be in that GDC Bio Portal instance that I'm showing you today. Another question. Does GDC Bio Portal have tools to automatically generate comma plots for these data? I think I would have to take a look at the GDC Bio Portal documentation. If you want to create a support ticket, we can look into that. Okay. I think that's all the questions. So I just want to thank everybody again for attending. I hope this was useful. There will be additional workshops in the future, as we have said. So please email the support email if you have any further questions. Thanks, everybody, and have a great day.